idea that some tropical plants like acacia trees need a little extra help defending themselves, and they will recruit ants in this particular case and feed them nice lipid rich, they're called Belchim bodies, that they produce on the tips of their leaves, so it's easy for the ants to harvest. And then all the different spines on the tree allow for some nice nest areas for the ants, so they can live there, they can eat there, and in return, they patrol the plant and help keep the plant pest free, especially from insects, of course. And it can be very effective. So that is a really interesting relationship because the plant has co-evolved with these ants to essentially have their own army of ants to help them. But that doesn't mean that plants are, unable to defend themselves. I think you've heard enough stories of poisonous plants already in your life that that certainly they have the ability to defend themselves through toxic chemicals. And so there's still a ton of research that needs to be done on this, even though there's already been a lot done. But, plants, if you think about it, because they're stationary, right? Wherever that seed lands and they germinate and grow, that's where they're stuck. And so they are faced with this idea of, I can't run away, I don't have a gun or any way to defend myself that way, so I have to deal with, okay, what kinds of things can I do if I can't run or can't fight with a physical mechanism? And so toxins, poisoning would be a good option. Even something simple as lignin, which is part of their natural body, is also a defense. So these, lignin is a very complex branched polymer. Even something like that, it's hard to digest. You have to have very special enzymes. Trichomes can be filled with glandular trichomes in particular with different secondary metabolites. We use some for essential oil extraction, but you can also find different toxins. There's even production of certain hormones that are found in animals. So the plant is making an animal hormone so that when that particular animal chews on the plant, it can mess with that animal's life stages. They can do it for insects. There's certain insects that they can do this to. And they can do it in mammals. So certain cattle, for example, have been known to abort if they're pregnant. So cows, female, because they're chewing on the wrong plant. And so if you are growing or raising animals for meat production or milk production or whatever, you need to be able to assess your grazing areas, which can be difficult if you have massive, hundreds of acres of grazing. So it can be challenging.
Definitely specialized goats that for eating juniper.
Yeah, because they can handle it, right? Yeah.
They sell pretty high, the juniper eating goats.
There you go. That's quite the niche. What do you do for a living? I sell goats that eat junipers.
Right.
There's also, they use goats for spotted knapweed out west. Because spotted knapweed is a problem, not just here. I know many students have got jobs during the summer, and that's their summer job, is to pull knapweed all summer. And so out west, though, the cattle ranchers, there's a video where they are interviewing a cattle owner and rancher, and he said, I never thought I'd be at risk of going out of business due to a plant. But that's how serious it is out there in some areas where they're kind of at a loss. And so there are goats now that they can put onto their property and have them try to eat the spotted knapweed because they can handle it.
Is there a chemical in the trichomes, or is it just?
Well, they have a very particular smell when you're working with both those plants. So I'm almost wondering if it's a chemical. But I haven't tracked that down, but I know that, because I worked with potatoes a lot and I would get irritated skin too sometimes if I wasn't careful. And I know like tomatoes smell really strongly to me. I don't know if that's the case with others, but so I'm guessing it's a chemical, but I don't know for sure. Sometimes trichomes in and of themselves are an irritant, just the trichome, because it's pokey enough. But sometimes like with stinging nettle, it's not only pokey, but actually, when you brush up against it, it's almost like an injection. It kind of triggers an injection into your skin. So,
Those, those little like spiky plants that lay flat and on.
yes,
Step on.
yes. Yeah, those are not fun in bare feet. So, there's all kinds of options that plants have, other than they can't run away and, bring out some kind of weapon. Mhm.
That's there's the plant that's like, it can give you chemical burns if you put like their chemicals on you and it's exposed to the sun and it's like really bad chemical burns. Cause I know they were a problem like near where I live. No,
Is it the poison ivy type idea?
Isn't it like the sap of a tree or something?
It sounds familiar.
person, but no,
Ooh,
Oh,
garlic mustard?
no, garlic mustard, you think that's it.
Yeah, I've heard, this is it's in there. I just can't pull it out. It's in there. I'd have to mull that one over.
But like, why would a plant have that drastic of a?
Yeah, and then like what you're saying with, and then when the skin gets exposed to sunlight, it intensifies.
It's
And
like really bad.
yeah,
I
right.
guess it's like how some snakes have venom so strong it could kill an elephant, even though they're using it to kill mice. Like they just evolve a protein that's that effective.
Yeah.
and
Yeah,
It happens that way.
yeah, I'll have to see if I can come up with that, but yeah, it's, like I said, the research is ongoing and it'll keep going for decades because plants, when you look at the huge amount of secondary metabolites that can get produced. And a lot of it is due to, they're constantly adapting to their environment. So there's always new chemicals coming along too. It's not like, they're constantly evolving as they're getting these different selection pressures in their habitat. And so they keep making novel chemicals in response to a changing environment. So, this last bullet point, we talk a little bit about that in Plant Fizz because of this idea that chemicals that are being produced by one plant. So if a plant is getting attacked, very often they will produce a volatile chemical that they will let escape through their stomata, and then nearby plants pick up on that chemical and it actually cues them to start defending themselves against the same insect maybe that's attacking that particular plant. And so the debate is still out there of, is this like a signal or a cue? Those two words mean very different things in biology. Is it a true signal where it's done on purpose to warn the neighboring plants, or is it a cue that a neighboring plant is just picking up on and capitalizing on? And that story is still being teased out and figured out, but they definitely could show that when that research was first published, people said, oh, no way, it can't be happening, but they've done really good, many published studies now showing that this is really happening. And it can be a volatile that's being sent out to warn neighbors that, I'm being attacked, get ready because you're probably next type thing. Or it can be underground through mycorrhizal fungi connections from root to root system. And so there's options out there. It's not always just volatile. So I'm not going to get too much into the secondary metabolites, but I would like you to know that phrase. So these are compounds produced by the plant, mostly in response to their environment, what's happening to them, often to attack, whether it be a fungus, a bacteria, a virus, a mammal, furry rabbit coming along, or a deer. And so terpenoids are often the class of secondary metabolite that are targeting insects. It's not 100%, you know, there's some gray area here, but alkaloids are often for mammals. And so for our taste buds, it's a very bitter taste. Alkaloids are generally targeting mammals. And then glycosides, there's a range of different glycosides that can do kind of all kinds of organisms that are attacking the plant can get impacted by that. And so glucosinolates are famous in the Brassicaceae family. So if you eat broccoli, you know, that very kind of pungent taste of broccoli, those are glucosinolates that you're tasting. And so this is just trying to give you an idea of some of the different groups of chemicals. You know, they can be volatile, but not always. Very often they're not. And there are even, you know, ways that a plant, they're looking more carefully now at how does a plant figure out, you know, we know that they produce these different chemicals to target different pathogens or predators. So how do they know? Like, how does the plant know I'm being attacked by a caterpillar versus a bunny rabbit? And they can actually pick up on that. And it's actually, they think a lot of it might have to do with what's in the predator or pathogens, herbivores' saliva. So as the caterpillar's munching away on the leaf, some of the saliva goes into the plant and the plant picks up on that and says, oh, I got an insect chewing on me. Oh, yeah. And then it starts making terpenoids in response, for example. Whereas if it's a bunny rabbit, they might switch and do alkaloids. And so they do have this ability to be that specific with what is attacking me. Because if you think about it, if the plant is producing all these chemicals all at the same time, and like, I don't know what's attacking me, I'm just going to produce everything, that'd be exhausting for the plant. It would deplete its reserves, its nutrients very, very quickly. And so this ability to really fine tune and really figure out what is attacking me is important. And then these secondary metabolites usually aren't made in the plant until it's getting attacked. Because again, it'd be too wasteful of their energy and resources, their nutrient resources. It's like, that's a lot of carbon and nitrogen and oxygen that they'd be using in order to make these compounds. So they really only wait until, okay, now I'm getting attacked, now I'll ramp up my defenses. And they have this really sensitive way. The plant has way more genes on its chromosome devoted to sensing its environment compared to us. We're kind of muted when it comes to our ability to sense our environment because we're so mobile. You know, we have the ability to be mobile and get away. And so we don't have to be as sensitive, and plants are way more sensitive in that regard. So here's some lovely plants that are poisonous. So foxglove, although we get digitalis from foxglove, which is actually heart medicine. So sometimes, you know, this idea that dose makes the poison, too much of something can kill you, but just the right dose and it can actually help you. That's very often the case with a lot of the drugs that we isolate from plants. And so like I've seen foxglove plants in landscaped gardens around town every summer. Beautiful plant, but again, toxic at the right dose. Lily of the valley. My mom used to grow lily of the valley all the time in our front yard. But yeah, don't let your dog or cat chew on it. And so it's, you know, these types of ideas where you have to know your plant and be aware of that. Same with houseplants. You know, some houseplants that are for sale, they're toxic. You need to do a little bit of research before you, especially if you have little kids or pets. Often the latex oozing that comes out is very visible. And so generally you don't want to get that on your hands or ingest anything like that. And so you have something too like this that looks pretty, you know, it's a plant. And yet if you go and research it, you can find that many of these innocuous looking plants can be a problem. I think that one is dogbane, which isn't a lovely name, dogbane. Does that sound kind of sinister?
It's the bane of
Yeah.
like.
So plants are amazing in terms of the secondary metabolites. We talk more about that in Plant Fizz, but you know, you think about leaf tissue in general. Oh boy, we get all kinds of useful things from that part of the body of the plant. Certainly food. Rhubarb is actually the petiole that you're chewing down on or making rhubarb pie. All kinds of different herbs and spices, oils, of course. Lots of essential oils can be extracted from leaves or the trichomes. We're gonna try to do that after spring break. I'm gonna try to, I know that you have been trying to grow mint plants from seed, and I think I'm gonna give up in terms of trying to grow them from seeds. I don't know what it is. My mint in my yard is like crazy. It just grows so well. And we can't get it to grow in the lab here, so it's bizarre. Somehow we're gonna have to establish a big thing of mint and, huh? I think so, because where it's growing in my yard is very sandy and kind of it's not a good area, and the mint's like, yeah. So I don't know.
You have to be like, I hate you, live.
Yeah.
Growing.
Put negative energy toward it, and it'll go, yeah.
It thrives in that environment.
Has oregano and like mints, and I think I threw a lemon balm in there at some point, and we ignore it for like the entire year, and we look at it at the end of summer, and it's like a huge,
Yeah.
like bush.
Well, and they say, normally what they say is if you're gonna grow mint in your garden, you should maybe put it in a container because it tends to just overwhelm your garden. It'll, you know, it travels quite nicely and will set up camp throughout your whole garden. And mine is trying to grow over a sidewalk area and get into the next patch of grass. So it's, yeah, but for some reason we can't grow it. This is what, the second or third year we've tried to grow it from seed. And we can't get it to work. We tried to be meaner this time, like
Sand.
sandy or soil that we tried to mix it in, and we're trying, but it's not working. So give some thought to economic importance. Lots of options here. Certainly the drug list is ongoing and extensive. Even carnauba wax to wax your car. Agave, of course, is probably $1 billion industry just for the tequila. Symbols. So leaves are used in symbols. Can you guess? Yep. Toronto Maple Leafs, the hockey team. And the Boston Celtics, the basketball team. And of course, the flag on Canada's flag. I guess I should have put that there too. But anyways, so yeah, lots of options. Essential oils, wormwood. Over the centuries, wormwood has had a rich history of things that they claim wormwood can do for you. So these are some of the ideas. If you look in the notes section, it talks a little bit more. Absinthe, you might have heard about that. If you're over 21, you might have sampled a little bit of that, but very high alcohol content. So be careful. But so it says wormwood contains the chemical thujone, which can cause seizures and death in high concentrations. Thujone is a GABA inhibitor, but there's not enough thujone in absinthe to kill you or make you go into convulsions, but the high alcohol content might kill you. So be careful. Emmenagogue is for menstrual discharge, cholagogue, flow of bile. I mean, you look up some of these words and it's a long, long history. Tea, of course, billions of dollars per year for tea. Tequila, digitalis, cannabis, of course, which is, not just the THC or CBD. You have all kinds of other products that can be useful in different parts of the plant. So carnauba wax, palm tree, surfboard wax. Yeah, don't think about surfboards much in this area. Here's a good one. speaking of fabulous leaves, this is Bokila trifoliolata. This is the one I was trying to think of weeks ago, and I couldn't think of the genus name. I remember the species name, but so this is the one that can mimic other plants around it. In one lifetime, it can morph its leaves to look like plants around it. And so this is something that it does on the fly as it's growing. And they've done all kinds of cool research with this. And again, when this was first published, it was a small little like page and a half publication in a journal. And people immediately attacked it and said, no way is this happening. And then they did a little bit more research and they're like, oh, well, I guess it is happening. And so it was met with a lot of criticism initially, but then they did more careful in-lab studies where they had proper controls and they could demonstrate that this is happening. So it actually changes the size of the leaf, the shape of the leaf, the color of the leaf, and even how obvious the veins are, when you look at it. And they think that the plant is doing this to better camouflage itself so that it doesn't stick out in relationship to other nearby plants, with the idea that there's probably less chance of getting chewed on. This is a tropical plant, so there's lots of tropical insects that are busy eating leaves. And so this is, so the V is the Bokila, it's a vining plant, so V for vine and T for tree. And so, I mean, just look at how similar, you look at the leaf design between those, how similar they are. So long and skinny in figure A, but figure B, they're more short and roundish.
This brings back up the idea of plants can see and.
Yep.
They even bad.
Yep. So this is one of the papers that some scientists, plant biologists. So there's a field of study called, I don't know if they still call it this, but initially they called it plant neurobiology. But that was met with a lot of criticism. So they might have changed that phrase, but basically they're pointing to the idea that they do have their own version of sight. Not like us, of course, because we have eyeballs. They don't have eyeballs. But if you look at the cellular, what's happening at the cellular level in order for sight to occur, they have a version of that. Bacteria have sight. Algae have sight.
Do they change the vein structure as well?
I don't think they can go like from parallel to netted. I don't think they can do that. That's a good question though. Because most of this looks parallel. Mostly. I've not noticed a netted venation in all there's not a whole lot published on this yet.
So to identify them, do they have to use DNA or?
Yeah. Yeah. I mean, they know where its native habitat is. But yeah, I mean, it's like, if they really want to be sure, because it's such a good mimic, they'd have to do DNA sequencing. So they think phytochromes might be involved, where it's sensing, helping it to sense light versus dark patches too. And I mean, there's all kinds of ideas. It's very controversial research right now. There's a lot of controversy over this, but I mean, proof is in. I mean, just looking at this, it's amazing. They're doing something. I believe they did. I'd have to check again how they assessed that was truly Bokila trifoliolata. They must have done something like that. So, this idea that in real time during its life, and even like, so it's a vining plant. So they've done experiments where they have one part of the vine on one type of tree, and then they have another tree established with different leaf patterns. And on the same Bokila vine, you can see two different leaf patterns, one mimicking this leaf on this tree, and then further down the vine, those leaves are mimicking the second tree's leaves. And so even on one plant, it can do different mimicry. And so it's just amazing. And so it's very rare, more animals do this mimicry idea than what we know of in plants. But then again, maybe we just keep missing it because there's such good mimics and we're not picking up on it because we're not doing the DNA assessment of some of these plants. So it's kind of interesting to see. I hope they'll continue with this idea and see if there's more plants out
There's
there.
Because it's like there are butterflies who look like poisonous butterflies. And
Right.
that look like poisonous
Right.
monkeys.
Right, so it's like, it's not shocking, now that they've did a really good example with this one with their study on it. It's like, well, there's probably more out there. We just are kind of blind to it. We're not picking up on it. So anyways, leaves are amazing, but in terms of the plant body, probably one of the more, well, it is, the most susceptible to attack. And so when you are looking for secondary metabolites, we tend to focus in on the leaf tissue. But, that's really the large source of all the chemicals and drugs that we used, today and with human medicine and animal medicine. And then, of course, leaves give us much beauty up here in the fall. Beautiful to look at, lay down in the woods and look up at a beautiful fall forest. And the great views you can get when you're up on Sugarloaf and so on. So lots of beauty there as well. Okay, so remember now, this second slide set will not be on the exam on Friday. We're saving that for the third exam, okay? But let's go into the exam material and let's stop here with that. And so the next topic that we will get into after break will be plant tissue culture and biotechnology, an introduction to those concepts. And those slides have been posted already. And then there's a very short slide set on plant breeding that the book does a good job with. And so I created some slides as kind of a study guide for that section in the book. So make sure you read that section on plant breeding, but I'm not going to go through it in lecture. But I will go through the tissue culture and biotechnology. That one's a little bit needs more explanation because there's some detail in there that you'll need explaining, okay? So, all right, but let's think about the exam. We have, starting with the photosynthesis slide set, and then all the why it does that. Oh, and then I did look at your quiz in the lab that you took that closed today. And so I wanted to talk a little bit. There's a few questions in there that versions of those will likely show up on the exam. So I want to make sure we're clear on those as well, because I know he has to hand grade that, so you're not getting your answers right away. Questions on anything to do with photosynthesis? Remember, the bulk of the material is, we're just kind of touching on the light reactions, and the bulk of the material is the Calvin cycle, C4, CAM, the PCO. So, but what are some lingering questions with photosynthesis? So I've been asking like Google form questions, or we just are looking at the PowerPoints and really trying to zoom in on a lot to deal with Rubisco, because there's some issues with Rubisco that you need to think about, and that can have profound implications for what happens to the plant. And it seemed like when I looked over the Google Forms and and the assignments, seems like we're on track with that. So, you know, but the idea there that you have to know the difference between your 5-carbon sugar versus Rubisco. And so looking at the CO2 coming in, reacting with RUBP. So it's in the active site of Rubisco, which is that sloppy enzyme. Let me go back to this really quick. Questions on the C3 cycle? Yes.
Ducks. Table two.
So when you think about the Calvin cycle, definitely the first step you got to know, because that's what kicks it all off. So that very first step, whoops, I should have gone a little bit more. When you go, you know, react RUBP and CO2 in the active site of Rubisco, and then you end up with PGA. So that first step, very important. Thinking about too, when you look at this cycle, like, you know, the huge goal of the light reactions is to get the necessary molecules to make sure the Calvin cycle can go, right? So at the very first step, you don't have any ATP or NADPH being used, but then you do see there's other steps in the pathway where you do have. So sometimes this pie chart is a nice example of where you're kind of seeing what's happening with where are the products of the light reactions getting used. And so carboxylation, the first section of it, there's no input of ATP or NADPH, but then the next two phases, you definitely do have to use those products of the light reaction. So here, you know, if you go from the CO2 and the RUBP reacting with Rubisco, you get your PGA. And then phase 2, knowing that you get this PGA going to the G3P with input of help from the light reactions. So this is a nice figure to study because it kind of summarizes the big picture ideas on the Calvin cycle. And then here they're showing that the glyceraldehyde 3-phosphate is a branch point that once enough of that gets, we call it a pool. Once you get a big enough pool of G3P, some of it can get sent off for not glucose, but sucrose. Okay, sucrose synthesis or starch if they have plenty of sucrose already cooking along. And then the last step is to make sure that you get enough of RUBP up and running. And so they have to use a lot of that G3P in this last step in order to regenerate RUBP. There's a lot going on here, but we're not getting into that in this class, okay? There's many, many, many steps here, but I think this pie chart is a nice, for this class, this is a nice study tool because it kind of gives the major points, not so much the detail. So studying figures like the roots, stems, and leaves, the figures that are showing up in the PowerPoint, the lecture PowerPoint, like those are, study those, they're going to show up. Some of those are exact photos, figures are going to show up on the exam. So pay attention to, you know, when we looked at root anatomy, stem anatomy, the leaf anatomy, make sure you're looking at those pictures and figures. Other things on this slide set. about the PCO? Go back to. Ah. Anything, anything lingering with the PCO? Good thing you don't have to memorize all that. What a mess.
Maybe so many things.
But again, the kickoff, I do want you to know what kicks it off. That's a really important concept. So here you have that idea that too much oxygen is around Rubisco, and Rubisco's like, yeah, I don't care, I'll take oxygen. And then you get that issue with RUBP reacting with oxygen instead. So that's being shown here with oxygen coming in. Whoops. So that's right there. And also over here, they're showing RUBP and oxygen. So that assignment that that you worked through was going through. So your assignments, the right answers should be posted that you have access to. So make sure you go back over those assignments, because those questions or something very similar to that is going to show up on the exam. Okay, I made that homework not to keep you busy. It was to help you learn for the exam. So knowing that there's 3 organelles is important and what they are. And that they say this is a loss of carbon. Okay, because the plant is actually giving off CO2 in the mitochondria, which is a problem. And so if you force a plant to run the PCO cycle, it doesn't do well compared to a plant that can eliminate the PCO. So they consider it's a loss and extra energy and resources that are going the wrong way. Other questions on this slide set? We'll make sure with the C4 and CAM that you're not confusing the two. C4, we say it's a spatial separation of what's happening. You have 2 different cell types and things have to vector around, move around. But CAM, it's all happening in the same cell, but you have a night-day rhythm of what's happening. So make sure that you thinking through that. What about root stems and leaves? Are there any issues? Once again, it's the lecture content, and what we talk about in lecture is the most important. So anytime I'm asking you questions in lecture, like, is this a leaf that spends more of its life vertical or horizontal? I mean, when I'm going through that with you, that's like important stuff. I want you to think about that. So anytime I'm asking you questions as we spend time together, I'm trying to alert you, this is something to think about. And then, we talked about how can you tell the difference between a root versus a stem in cross section? How do we look for that? And so these types of ideas, we went through that in class. I think we did some Google Forms. I believe Duncan also did work that in a little bit to his lab quiz. Oh yeah, and he's talked, what's the difference between a bulb and a corm? So a bulb, onion bulb, what is that mostly made out of? Leaf tissue. And then the corm is mostly really hard stem tissue, right? And then he asked a question on secondary growth. Secondary, I think it must have been a true false. Secondary growth occurs only in monocots. False. That's true. I mean, you're correct. False. What else? Oh, and then he asked you some monocot dicot questions. Visually.
Label that.
Yeah.
We're gonna have to know like every part of like those four
Yep,
parts.
so know these zones. And then, don't be surprised if you might see something like that.
First monocot, so,
Mhm.
okay.
Root versus stem, monocot. So these are things I asked you, right, in lecture. So these are things I consider relevant and important to think about. Kind of fun too. Once you learn the tricks, it's like, oh, whoops. It's and then let's see. That's about it, I think. So what, do we have any questions on roots, stems, leaves?
Yeah. It is- I was looking at the specialized one, it's like because they were labeled differently, they were, is a stolen a runner root? Is that just a fancy word?
So stolen is essentially an above ground modified. So when you, did I put that in here? I know, did I put that in here? Because I had, where is it? Stems? Where did I put that? Maybe in the, I know, but I put something new in and I wanted to show you all. Did I do that? I don't think I did. Was it in there? Ah, oh, I'm losing my mind. I know, because I grabbed something off of the internet and put it somewhere because I wanted to show you, because I've seen different definitions of a stolen and I wanted to clarify. So a stolen, the way I define a stolen, it's an above ground modified stem. Okay, above ground modified stem. That's horizontal across the ground. So strawberries are a really good example of that. They send off these stolons to, and then they pop up new plants off of it. Whereas a rhizome is a modified underground stem. Okay, underground, a rhizome goes underground. And then a tuber, like a potato, that's actually a modified rhizome that's been modified, for nice plump storage of starch and proteins. But I've seen some sites garble up what a stolen is. They, in my opinion, it's an incorrect definition. So I just wanted to, I thought I put it in here. I'm losing my mind. I did grab it and put it somewhere. Yeah, and then I looked it up after we talked about it because I wanted to, like, I don't know what I did with it. It's in there somewhere. Leaf litter, it can be, but that's still above the soil level. You know what I mean?
The strawberries, they're just like. out there.
Yeah, strawberries, you can usually see them. Yeah.
Plant
See, plants
ecology,
are
we
holding
have
it.
that dig for sunflower.
Mm, yeah, that I can understand in a forest system. Yeah, yeah. And you might encounter somebody who will tell you a different definition of stolen. But follow mine for this test.
Yeah, that was the main thing to clarify.
Yeah, yeah. Anything else? Roots, stems, leaves? I will be checking email into the evening. If you do have any questions, please make sure to reach out and then come prepared on Friday. This is a bigger exam than the last one, the first one. It's slightly over 100 points. Okay, so. See you Friday. Yeah.
And that- Oh,
Hang on just a sec, let me. Oh.